In this video, we will discuss about the mechanism of CHIC2 STUB1, how they interact to control the cell surface cytokine receptors. Mainly this IFNgR1 and IL-27R-alpha. So in the previous video, we have understood that there is CHIC2 and STUB1 can interact with this cell surface cytokine receptors and helps in regulating them. For example, if this CHIC2 or STUB1, either of them is knocked down, then they will see high expression of the cytokine receptors. Since the STUB1 has a role towards this E3 ligase, they hypothesized that the CHIC2 and STUB1 might form a complex and that interact with the cytokine receptors, and instead of expressing them in the cell surface, they divert them towards ubiquitinations. So they need to validate this hypothesis. So first they started with checking the mRNA levels in IFNgR1 and IL-27Ra in the STUB1 knockout and the CHIC2 knockout, and they did not find any changes in the mRNA levels, indicating that there is no changes occurring in this at epigenetic levels. Thereafter, the hypothesis that most of these proteins, mRNAs that is converted into the proteins, will be diverted towards ubiquitinations. So they form a knockout where they knock out this U-box region that prevent the ubiquitination process of this STUB1. So by knocking out this U-box region, they can expect to form they are expecting to get a more amount of IFNgR1 and IL-27R-alpha, because ubiquitinations will not be occur in this STUB1 in which this U-box region is knocked down. First they check the formation of STUB1, and from this PDB protein analysis protein structure 3D structure analysis, they find out that this STUB1 without U-box are able to fold properly, and they will not be like reduced in the their segregation amount will not be reduced because of the ER quality control. And here they validated that hypothesis that in the case where STUB1 has been here they validated their hypothesis in panel D that when the STUB1 is knocked down and they are retransduced with this U-box region trimmed out, they are able to see more STUB1 compared to the wild type. And when they check it, the IL-27R-alpha and IFN gamma R-alpha amount using Western blot, they are able to see that its amount is high compared to the wild type, indicating that these ubiquitinations of this process of this STUB1 might be interacting with this IL-27R-alpha and IFN gamma R1 and diverting them towards ubiquitination. So without these ubiquitinations of this STUB1, they are able to see more IL-27 and IFN gamma R1 compared to the wild type. Thereafter, they wanted to check this IFN gamma R1 and IL-27 further by preventing these lysosomes pH change inhibitors, or preventing the lysosomes to function properly using this bilifomycins A1, and in that case also they are able to see more amount of this IFN gamma R1 and IL-27R1, indicating that it's the lysosomes degradation is a lysosomal degradation process that is causing the decrease in this IFN gamma R1 and IL-27R-alpha. Thereafter, they made like two hypotheses to validate this using whether what role this CHIC2 plays. For that they made two hypotheses. First one is that the CHIC2 might be interacting need to be present on the membrane, thereby it brings this STUB1 and the IL-27 and IFN gamma R1 close together so that the ubiquitination process might begins. So in order to check the hypothesis, they remove these palymitoylations using the mutation C6 with S, and then check whether it will have any impact on the IL-27R-alpha and IFN gamma R1. And the next hypothesis is that the ubiquitinations in the CHIC2 occurs at this lysis 117. So when they mutate these things, they should we make this CHIC in a form that it will not undergo these ubiquitations. So they wanted to check whether this ubiquitation of the CHIC2 is responsible, or the its presence at the membrane site is more important. So when they check the results for the IF and gamma R1, they are able to see that in the case where the no mutations of this CHIC2 occurs, they do not see any changes compared to the wild type, both for the IF and gamma R1 and IL-27 alpha, indicating that this giving us the idea that these ubiquitations of the CHIC2 does not play a role towards these cell surface cytokine control, whereas when this pelletolations region is knocked down, that is CHIC2 is not present at the uh, membrane, at that time they are able to see increased value about the IFN gamma R1 and IL-27R alpha. So this indicates that CHIC2 must be present at the membrane, and then it can interact, or it helps in bringing the STUB1 and these cell surface cytokine receptors, that is IL-27R alpha and IFN gamma R1, thereby initiating or promoting this ubiquitation process. This is what they have finally concluded from their results, or finally they come up to the logical conclusions from the observations they got in these results. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.